Hi, my name is Colin Gould, and I'm a 2021 Arnold Beckman Postdoctoral Fellow at Princeton University, where I work in the lab of Professor Dave McMillan. I feel very fortunate to have received a Beckman Fellowship, and it's impacted my career in several important ways. First, the independent funding provided by the Arnold and Mabel Beckman Foundation allowed me to pursue creative research directions during my postdoc at Princeton, without needing to worry about whether or not this research was funded by existing grants in my mentor's lab. This independence allowed me to grow as a scientist, and I believe that has prepared me well to lead my own research group in the future. In addition, the Beckman Fellowship has introduced me to a network of talented scientists, ranging from other postdoctoral fellows to undergraduates and faculty members. Whether it's through the Beckman Symposium or funding to attend other conferences, I've had many opportunities to make connections with other scientists, connections that I can rely on for advice, collaboration, and perspective as I continue on in my career. Last, the ability to transfer funding from the Beckman Fellowship to my next position, a position as an assistant professor at the University of California, Berkeley, will help to smooth this transition, and it will allow me to get my own group up and running more quickly. In sum, this fellowship has been an incredible opportunity for me, and I'm grateful to the Arnold and Mabel Beckman Foundation for sponsoring it, as well as for all of the work that they do to promote science more broadly. Now, I'll tell you a bit about my research over the past 3 years. My research at Princeton focused on photoredox catalysis, which is a strategy in synthetic chemistry wherein the energy found in visible light is used to convert widely available feedstock molecules into value-added products, such as medicines, agrochemicals, and polymers. The strategy is enabled by photocatalysts, such as ruthenium trisbipyridine, shown at the left-hand side of the slide. When a photocatalyst absorbs a photon of blue light, it enters a long-lived, high-energy excited state. This excited state is both a potent oxidant and a potent reductant, as well as capable of energy transfer up to 50 kcals per mole. As a result, an excited state photocatalyst can interact with common organic functionality or reagents designed to activate them, things like alcohols, carboxylic acids, and even carbon-hydrogen bonds, and convert them to the corresponding high-energy radical intermediates. These radical intermediates can then be interfaced with transition metal catalysts to form a wide variety of valuable chemical bonds, including transformations like amination, fluorination, and isotopic labeling, all of which are highly desirable in the pharmaceutical industry. To summarize, photoredox catalysis allows medicinal chemists to take abundant vegetative feedstocks and convert them to value-added products, increasing the speed at which they can test a wide variety of structures for biological activity and increasing the diversity of chemical structures that they can access. One thing that's missing from the toolkit of photoredox catalysis and synthetic chemistry more broadly are methods for the formation of sp3-sp3 carbon-carbon bonds. Indeed, the vast majority of reactions that form carbon-carbon bonds require one of the partners to have sp2 hybridization. Within the class of sp3-sp3 cross-coupling, the formation of quaternary carbon centers, that is a carbon atom bound to 4 other carbons, remain especially underdeveloped and represent a long-standing synthetic challenge. This stands in contrast to what would be beneficial to medicinal chemists. In fact, it has been shown that the fraction of sp3 carbons in a drug candidate correlates with success all the way from discovery to market. In addition, quaternary carbons are associated with metabolic stability, conformational rigidity, and selective binding to proteins. Ideally, we'd have more methods to incorporate sp3 fragments and quaternary carbons into drugs. The reason that there aren't more methods for this comes back to the fundamental mechanism employed in most cross-coupling reactions. The interaction of a metal catalyst with alkyl fragments typically results in fast side reactions, such as beta hydride elimination shown here, which outcompete the desired carbon-carbon bond formation. To summarize, methods for sp3-sp3 cross-coupling and quaternary carbon construction in particular remain underdeveloped, but these methods would be of significant utility to medicinal chemists if they were available. My research in the McMillan group focused on a new approach to carbon-carbon bond formation, which we term radical sorting. In this strategy, 2 substrates are converted into the corresponding radical intermediate via the action of a photocatalyst. If these radicals were to encounter each other in solution, you'd expect statistical recombination in addition to a wide variety of other side products, resulting in only a very small yield of the desired cross-coupled product. However, the McMillan group has discovered catalysts, such as the iron porphyrin shown here, that can selectively sort these radicals, resulting in selective formation of a carbon-carbon bond. For instance, the iron porphyrin shown here can directly bind the primary radical at left due to the greater strength of the iron-carbon bond. This leaves an excess of the tertiary radical in solution, which can then interact with this primary iron alkyl species to form a carbon-carbon bond via bimolecular homolytic substitution, or SH2, the single electron analog of the classic SN2 mechanism. Notably, this SH2 mechanism is actually found in nature in the biological methylation mechanism of methylcobalamin, as well as in the mechanism of P450 metabolism. This radical sorting mechanism allows the formation of only the desired cross-coupled product, suppressing most other common side reactions. In my first project in the group, I worked with Wei Liu and Marissa Lavanino, co-workers in the Macmillan lab, to validate the proposed mechanism shown here via a variety of experiments, including isolation of the key iron alkyl intermediate. Once validated, this radical sorting mechanism was incredibly exciting to us because it offers a functional group agnostic way to form carbon-carbon bonds. What I mean by this is that we could potentially take any abundant functional group, any functionality that we know how to convert to a radical with photocatalysis, and we can use it now to form a carbon-carbon bond. Over the past few years in the Macmillan group, we've demonstrated that this is indeed true, and the mechanism of radical sorting has been used to cross-couple all the diverse functional groups shown here. A variety of sorting catalysts have also been discovered during this process. I've worked on 3 of these projects, highlighted here, and overall, it's been a very exciting time to be in the group. I'd like to end by acknowledging all of my collaborators on the work shown here and the Macmillan group generally. I'd also like to thank the Arnold and Mabel Beckman Foundation again for supporting this research. The Beckman Postdoctoral Fellowship has been an incredible opportunity for me, and I'm very grateful for this support.